Good afternoon, everybody, and welcome to the 2024 COVI Lecture Series. I'm Debbie Ingalls. I'm the Director of COVI and proud to be your host here today. So I'm pleased to introduce our speaker today, which is Dr. Sudarsana Poojari. Sud is a principal scientist here at COVI and also an adjunct professor in the Department of Biological Sciences at Brock University. Sid obtained his PhD degree in plant pathology from Washington State University and then served as an NSERC postdoctoral fellow at Agriculture and Agri-Food Canada's Summerland Research and Development Centre in British Columbia before joining us here at COVI in 2018. Sid's main area of research is in grapevine viruses, and as Canada's expert in grapevine virology, Dr. Poojari serves as a scientific advisor for the Canadian Grapevine Certification Network, or CGCN. He developed molecular diagnostic methods for virus detection in grapevines that were validated by international research institutions and are recommended for quarantine and certification programs. He is currently the academic lead for a Genome BC grant, also working with the Canadian Food Inspection Agency, or CFIA, with Mike Rott as the scientist there, to develop diagnostics for grapevine viruses based on high throughput sequencing. An exciting output from his work with CFIA and CGCN, along with Ontario Grape and Wine Research Inc., is the establishment of a grapevine germplasm repository here at Brock University, the core of a domestic clean plant program, and this is only the second one in Canada, with the first one being held at the CFIA facility in Sydney, British Columbia. So Dr. Poojari's research interests also extend to disease management, virus-insect-host interactions, landscape genomics, as well as just acknowledging all of his publications, 22 peer-reviewed articles, 2 book chapters, and a number of presentations and technology transfer activities that he's currently doing as the principal scientist here in COVI. So please join me in welcoming Sid to speak to us today about the importance of clean plant programs around the world, but especially here domestically in Canada.
All right, good afternoon. Thank you, Debbie, for a kind introduction. Just jumping in, this is the outline of today's presentation. By the end of the topic, you'll be introduced to some of the grapevine virus diseases to start with, and first trying to learn why it is important, or what are the different virus diseases, what they cause, what type of economic losses they cause, for example, and also when we know about different viruses that cause these different virus diseases on grapevines, we will be knowing how to test them, especially in terms of how clean plant program works and how to eliminate these viruses in a given grapevine host, for example. And also learning about the grapevine certification standard, how the structure look like, and how different they are in terms of different countries, different regions, how they practice their clean plant programs based on what reasons, for example. And also we will be knowing about what we learned so far here in Canada in terms of clean plant program for grapevines and how it is prospering further. So to begin with, the importance of the grapevine viral diseases. I know you might have heard from many speakers who works on grapevine viruses or grapevine pathologists, even from my previous lectures, that we do have issues with grapevine viruses, not just here in Ontario, but wherever the grapevines are grown in the world, the viruses or viral diseases are one of the biggest concerns as far as the industry is concerned. Some of the main 4 or 5 major diseases I will name here: grapevine red blotch disease caused by grapevine red blotch virus, grapevine leaf roll disease caused by a number of grapevine leaf roll associated virus, and among them leaf roll associated virus 3 is one of the most important virus because of its widespread and how negative effect it causes on the grape growth as well as the wine quality. Other diseases to concern is the grapevine family, grapevine phenogry, virus diseases. There are a few other diseases of less concern, but still would make a strong impact, especially when you come to the rupestris tempeting associated virus, as well as other witty viruses that are mainly associated with incompatibility issues when grafting, because we know that we cultivate these grapevines, we use the grafted vines. That means we need to have a rootstock and scion, and if some of these viruses are involved, especially the witty viruses and rupestris tempeting associated viruses, we do have seen evidence of grafting incompatibility. That means success rate of these grafts will go down drastically based on what combination of virus is present, of course. And most importantly, especially when it comes to viral diseases and grapevines, except for red blotch, as we know, only grapevine red blotch virus is the only causative agent for this disease. But viral diseases like grapevine leaf roll disease are always complex. That means they are associated with a number of viruses. So, without further emphasizing on how many diseases we have, which is always based on the region, based on where the vines are sourced from, most of these grapevine viruses are transmitted primarily through planting material. That means we all know that the grapevines are propagated through the planting material. So I would like to emphasize always that when you are using the grafted material, even if it is a rootstock that is infected with the virus here, which is indicated in the red dot, or scion that is infected, when you graft them, the daughter vine is going to have the virus throughout, because the virus can move either from rootstock to scion to rootstock. And in case of wound-rooted vines, if you take cuttings from the mother vines and try to propagate, obviously the daughter vines is going to have the virus. This is the primary transmission. You can also ask, like, what about the secondary transmission? They are based on the type of virus species that is infected, that is being infected. We do have evidence of different insect species or nematodes act as vectors for these diseases. For example, leaf roll, most of the leaf roll viruses except the leaf roll 2 are known to be transmitted by a number of species of mealybug as well as the soft scale insects. When it comes to the red blotch virus, although there is a lot of research going around different regions, but there is an evidence from California that alfalfa three-cornered alfalfa treehopper is a known vector for this particular disease, which we don't have it here in Ontario, but there is a lot of research that is going on from the AFC Vineland that we are trying to look for possible insect species, including the tree hoppers as well as the plant hoppers. So let's dive into to know what is going on around the world in terms of the clean plant programs are concerned. So the first thing that comes onto the table is the National Clean Plant Network in the United States of America. So although they are a bit far ahead in terms of establishing this clean plant network, that means having different clean plant centers all over the United States in different states, but also having a framework of between the federal government agencies as well as the state government agencies as well as the academic institutions for research purpose. So for example, here, as I mentioned, the US Department of Agriculture is investing over $700 million in 374 projects. Just this news has been announced a month back for Plant Protection Act section under 7721 program. I know there is a lot of text here, I do acknowledge that, but I just wanted to point a couple of things here that, for example, close to $6.2 billion allocated to California and Florida for nationally to support detected dog team training and maintenance of domestic pest detection. Why I want to emphasize this is the National Clean Plant Program is not just looking at one direction, it has to be a multiple ways in terms of how you are doing your national surveys for screening emerging pests and pathogens, for example, how differently you are approaching based on what type of large-scale infrastructure you are looking. For example, citrus industry, which is very large. If you want to screen the entire citrus industry in the United States, in the states like Florida and California, you need to have different approaches to screen a large number of orchards, for example. And also another example is the stone fruit and orchard commodities. They are getting close to $1 million to support the pest detection surveys in the states like Colorado, New York, Texas, for example. And also the last one is the certified disease-free citrus planting material is getting close to $1.8 million just to protect American nurseries and growers from the economic losses caused by citrus plant diseases. So what is this the USDA is doing? So they say that they have a network of clean plant centers, as indicated in this map here, with different colored dots representing different crop commodities, for example, berries, citrus, fruit crops, grapes. You can see how diversified they are based on what type of crop grown in each state, for example. And among these, for example, California is one of their main centers for the clean plant network, especially when it comes to grapes. And the good thing is, in 2014, through Farm Bill, they made this National Clean Plant Network a permanent program with dedicated funding, so that whatever they have built, they have to be maintained throughout, helping both growers as well as nurseries. So when you look at the numbers from the USDA website, you can see I have listed different crop commodities, fruit trees, number of cuttings, number of birds, close to 500,000 cuttings. When it comes to grapes, there are clean plant networks that are spanned close to 6 different centers in the United States. They maintain over 1,000 grapevine selections and distributing over 700,000 cuttings and buds. And same goes to the berries, one of the biggest crops as well, close to 30 million plants produced annually. Citrus, more than 100 citrus selections are being maintained and also distributed close to 600 million trees annually. Hops, they also give importance. Sweet potato is one of the success stories as well. They maintain over 170 sweet potato accessions. And also it reaches to horticultural crops like roses. They do have maintained 600 different selections in their clean plant centers. So when it comes to the grape, narrowing it down, as I mentioned, there are 6 centers. First, the main center being the FPS, Foundation Plant Services in UC Davis. And there are other centers in the Pacific Northwest, especially in Prosser, Washington State, in Florida, Missouri, Cornell, which is close to us, and also North Carolina. So I can come to this a little later. This is how the Foundation Plant Services structure looks like. But when you say, okay, we do know about the importance of clean plant network, but what is happening in other countries? So if you take an example like emerging countries, emerging economic countries like India, they recently in 2023 in their union budget announced that they're going to invest close to $196 million to establish 10 different clean plant centers across the country. So that tells the importance because when you look at the changing geopolitical situations, especially after the pandemic, people are concerned, especially the countries are concerned about their food security. So the clean plants are playing a very important role, as like any other countries, say like United States of America or Canada, even India also, if any new crop clone variety has to come into the country, it would still has to go to the quarantine programs that are designed by each countries as per their regulations. It takes 2 years to be released to the growers, for example. So through these clean plant centers, they are hoping to achieve, to reduce that timeline from 2 years to 6 months. So they are investing a lot on crops like apples, walnuts, almonds, grapes, and obviously mangoes and pomegranate. If you can, why it is happening is because if you look at the import numbers of Indian fruit market, for example, especially apples, almonds, and grapes, their imports has increased close to 40% from the year 2017 to 2023. So you can see the importance of having these clean plant programs at their disposal. So when you look at the other countries, much smaller countries like New Zealand, especially focused on grapevines, they do establish their grapevine certification standards because they have done a bit of their homework and what type of virus issues they have. They do know that they have their main issue is grapevine leaf roll disease. So especially the grapevine leaf roll associated virus 3, which can be transmitted primarily by planting material, secondarily by different species of mealybugs, which they do have, unfortunately. Their focus is to only give the importance to leaf roll 3. Any material that is being planted, they make sure that it is tested for this particular virus. And again, the clean plant programs depends on what type of exact scientific knowledge they have so that they can be best implemented to get the most benefit out of it. So if we look at the numbers, especially when it concerns to the grades, of course, most of these studies are coming from either states like California or Washington State or New York because of the sheer size of their industry and how much they invest in their research. For example, leaf roll disease and red blotch disease, as you can see, both are coming from a study by Ricketts et al. In New York and I think both in California and New York. You see the numbers, they can range anywhere between 61,000 to 558,000 per acre lost over the period of 25 acres. Of course, these studies are based on how much percentage of the virus that they have and what are the mitigation practices that are being followed. There are a lot of combination scenarios. For example, they are taken into consideration in coming up with these numbers. And also we do know that by different economic studies, Pierce disease on grapevines in California costing them close to $92 million annually. And a recent economic study came out that the screening of these grapevines only for leaf roll 3 virus is saving California grape and wine industry close to $90 million annually. So you can see the importance of this clean plant program. So now we talked about the importance. Let's see how we can test this virus and what are the different methods to eliminate that can be used in the clean plant programs. So grapevine virus diagnostics, there are different ways to detect a virus in a given sample or in a wine based on the symptoms. Sometimes it is not accurate because there are viruses that could cause a similar type of symptoms on a given type of grape varieties, and also they can be confused with other things like a mineral or the deficiencies of minerals. There are biological indexing where you can use, say for example, you have rootstock that is infected with a particular virus, and you can graft a scion that is healthy, and then try to establish that wine and see if the virus from the rootstock will slowly move to the scion and try to appear symptoms on the leaves, which is obviously time consuming because we are dealing with a perennial crop like grapevine. It could take close to two years to see any visible symptoms. And we do have like serological detection assays like based on ELISAs, mostly antibody-based assays, But the problem with this is some of the tests are really nice, they're very specific, but unfortunately antibodies are not available for a number of Grapevine viruses. And when it comes to the comparison with the molecular techniques like polymerase chain reaction, PCR, the lasers are considered less sensitive than the PCR. So the golden standard right now to use in the clean plant program is mostly molecular diagnostic techniques, especially PCRs or quantitative PCRs, like traditional quantitative PCRs, as well as digital droplet absolute quantification PCRs, as well as the new techniques like next generation or high throughput sequencing. We'll talk about this a little bit further. But recently, this high throughput sequencing technique has made its way into the Clean Plant program, especially getting approved in the National Clean Plant Network for Grapevines in the United States. And also there are efforts to include this internationally acceptable protocol. There are a lot of research going on around this area. So what's the difference between traditional and HTS? Is when you put it simply, PCR you can detect one virus at a time. High throughput sequencing you can detect all viruses at a time with more sensitivity and accuracy. So it is more comprehensive in the sense you don't need to do 30 different tests for, if you have 30 different viruses in your screening list of the certification standard, you don't have to do individual tests. You can just do one test. There are different types, especially when you're talking about like, okay, what are the different types of HTS? You can use the total RNAs, total nucleic acids. There are different types of viral enrichment process are available. I'm fortunate to work with Dr. Mike Roth on a Genome BC funded project where we are trying to develop to come up with a better technique to use this HTS in the clean plant certification programs. So that's the project that I was talking about. It's called Cleanse for Grapevine and Grapevines in Canada. So to summarize, what we are doing here is to come up with better virus enrichment protocols as well as sequencing protocols. Not just the sequencing protocols. Once you do the sequencing, you have a lot of data that is generated. How best you can analyze the data using the tools, bioinformatic tools that are available, which can be made available publicly so that it can make it cost effective. So one of the main aspects of objectives is to reduce the total cost that is also occur when you're doing the HTS. So these are different partners in this project. So we are working with the Canadian Food Inspection Agency, which is the receptor party in this project, working with the Canadian Grapevine Certification Network, and also being supported by the Genome BC and Genome Canada. We do have researchers from University of Quebec, University of Victoria, the University of Sherbrooke. So I just want to tell you, okay, it's just not one method, but the detection techniques will keep on evolving, especially when it comes to the virus diagnostic techniques. So I have a couple of slides that what we have recently published, going to publish a few of them, is using a different techniques called like CRISPR-Cas, using CRISPR-Cas, for example, to detect the Grapevine red blotch virus. It is published in 2019 from here in Rock University. So what we did here is use the CRISPR-Cas2a coupled with the gold nanoparticles to detect Grapevine red blotch virus up to the automolar values. So that is very sensitive in terms of how much we can detect. So we also trying to develop another amplification-free detection protocol method using CRISPR-Cas13, where you can see we are going up to 2 nanograms of Grapevine leaf roll associated virus concentration, and we are able to detect within 10 minutes. The only emphasis I wanted to make here is we keep on developing and testing these protocols so that we can compare different methods to see which is the best method in what scenario, for example, to whether you can use it in the screening of the viruses in the clean plant programs, or when you are doing for research phase, for example, if you want to identify a particular insect vector for potential vector capacity, you can use this kind of sensitive methods to find the virus. So it's also another virus, same droplet digital PCR assay for detecting Grapevine red blotch virus. Muti, who is a postdoc, is also working on this project. What he found out is it can go up to 10^9 dilutions of the virus concentrations when you're using this plasmid DNA as a standard. But also it detected that it can go to attomole concentration to detect even a single copy of the virus in a given sample. So that's about the detection methods. Let's see how the certification standards looks like. So when it comes to the clean plant program, there are different types of certification stages. So when it's anything that has to be included in the certification program, whether it is the cyan variety or rootstock variety, first based on what is the standard says, like your certification standard says that you need to test, say for example, 30 different pathogens. First you test them. If the particular material is tested negative, what we call it as a candidate material, which you can use to propagate, generate a nuclear stock, and then the nuclear stock will go into a propagation stock where you can use both rootstock and cyan to make grafted plants. And once you graft and establish these grafts, that's what you call the certified stock. Similarly, when something tests positive for a pathogen, so you go through a process called virus elimination. I do have a couple of slides on virus elimination. You go through it and then you test it again, and when it tests negative, it goes through the same process of nuclear stock, propagation stock, and then certified stock. So how do we eliminate the virus? So we do use the technique called apical meristem tissue culture for grapevines, which is an established technique considered as a gold standard for this for elimination of virus cultures. So why it is important when you ask that? So apical meristem in any plant species, it is very rapidly developing part of the plant. That means this particular the apical part of the shoot that if you take, that's where the cells are very rapidly multiplying. That means they don't have an established vascular structures, which is very much necessary for these viruses to move from plant cell to cell. So and also another important consideration is these viruses are obligate parasites. That means they depend completely on host cell machinery to either replicate or get into a certain type of structures using the host cell machinery. Since this apical part of the meristem is very rapidly multiplying, they don't have all the components. The protein synthesis is very limited there. So that's why viruses cannot able to establish themselves in that particular part. But when you come to a little below this apical meristem, maybe here you might see some viruses creeping in, but not at the top. So we don't know the exact mechanism, the molecular mechanism, nobody understood so far, but we do know that we can use that apical meristem by looking under the microscope, and by looking at 100x zoom, we can slowly cut that apical part of the meristem and then try to place it on a nutrient media to try to grow it, because it has the ability to grow into a full plant. So that's how it looks like when you look into the microscope. And Linzu, she's not here today, she is capable of cutting this apical part of the meristem and try to establish it in a full-grown plant. So I can't do that. So even when you look at international plant protection organization like European Plant Protection Organization guidelines, for many plants, they do show, okay, if you're working with the perennial crops, you can do the heat treatment, especially for grapevines, for canes of more than 2 years old. And meristem is very much recommended. Shoot tips can be cut at 0.4 to 0.6 micrometers long, and then you can inoculate them into artificial media, and that's how it looks like at the beginning. And then once they grow, maybe as 4 to 6 months old, they will look like that. And then close to 8 to 10 months, they will look like this. And then slowly, once they establish their root system, of course, in this different process, we use different type of nutrient media, different type of hormones to generate, to multiply the shoots, to multiply the roots. So different steps are involved. And once they establish something like this, we think that they are able to go into a more natural type of environment. We transfer them into greenhouse conditions and grow them like this. And then slowly they're developed into grapevines. So to summarize, as you can see, any virus-infected grapevine that has to be included in the germplasm repository, whether it is coming from a virus-infected plant or some other source, it doesn't matter. When it has to enter into the germplasm, it has to go through the same process. So we do the ethical meristem tissue culture, generate the plants, test it with the high throughput sequencing, and then slowly move them, establish it, and slowly move into a greenhouse or greenhouse conditions, and then we'll establish a mother block. And typically, in any mother blocks that are there in the clean plant centers as well as in the nurseries, they have to follow the certification standards as designed by the particular authorities, where mostly you have to do audit testing for the pathogens of concern, as well as monitoring for the insect vectors, for example. So different certification standards, different certification agencies, they have different standards. We'll go through them quickly. But to summarize, this is how it looks like. So then we can ask, what is certified grapevines? So simply to put it to perspective, the Clean Plant Center acts as a source for this certified grapevines. They can provide a grapevine material, maybe not enough for the entire vineyards in that region, but to initiate the propagation by nurseries, for example. So the nurseries are the ones that has the job to scale up the production, taking this certified material from the certification agencies. So they multiply them to meet the needs of the grape growers. And then certification ensures the quality. Nurseries must follow, for example, the grapevine certification standards here in Canada. They have guidelines how to follow the pest management or virus testing to maintain that certification standard. And also standards requires frequent updates. As we know more about through our research, we do need regular updates for the certification standards. We go through those things in a couple of slides. So when you're considering the clean plant program, there are a few questions that are always asked based on the region. Even when you look at the states, even for a particular crop like grape, there are different type of certification standards for California, different type of certification standard for Washington State because they understand what are their pest pathogen problems, and they also work very closely with their regulatory agencies, both regional, state level, provincial level, as well as federal level quarantine regulations to make sure that they can adapt this certification guidelines according to their conditions. And also they would be asking questions like, okay, there are different type of viruses that can be infected. The grapevine can be infected, but they are all important. Can be we can tolerate any of them. Say there are some ubiquitous viruses that are there that are not reported to cause any economic damages. Maybe we can allow them because it's probably very hard to eradicate them. And how to adapt the latest diagnostic methods. We talked about this a little before, and also ensure this propagating material is available to how to ensure this material available to nurseries and growers. So, and most importantly, once you have availability of these clean vines that are planted in vineyards, and how to prevent the reinfection, how to prevent the secondary infection. So we do need stock clean and stay clean programs. So what we learned so far, Canadian perspective. So to go further before, first we need to understand what are the quarantine regulations in Canada when it comes to, say, like importing grapevine planting material from different sources. So this is the directive from CFIA G9434, which clearly said that Canada regulates the imports of grapevines from foreign countries through CFIA, the Canadian Food Inspection Agency. So it allows the import of grapevine planting material under US certification programs, especially from these 4 states: California, Oregon, New York, and Washington State. So other than US, CFIA do allow grapevine planting material from France and Germany with CFIA approved nurseries that are listed in the website, not all of them. So you need to make sure that you follow the guidelines. And then they do have a list in the same directive, like what are the different type of pathogens, especially the viruses, that are regulated. So you need to make sure CFIA has a mandate to make sure anything that is being imported, they do audit testing to make sure that that particular regulated viruses or pathogens would not enter into the states, into the country. So when you look at the National Clean Plant Network, especially for grapevines in the USA, they have a protocol called 2010. It doesn't mean that it is established in 2010, but they have been updating ever since. But since it is established in 2010, they call it 2010. But they do have a requirement of testing different viruses, 30 different viruses through different techniques, as we just spoke about, PCR, qPCR analysis, as well as now the high-throughput sequencing is approved for provisional quarantine release to fast-track this process. So as mentioned here in this little graph, the FPS has the foundation block, which is tested every year, and then the nurseries have registered blocks from FPS, which has to follow the certification standards that are guidelined by state certification programs, and then from there it goes to the fruit production vineyards, these growers. So what do we have? We do have a Canadian Grapevine Certification Network established in 2018. The long-term certification standards, as mentioned here, any new varietal selection that is coming in, if it is tested negative for all the pathogens that are listed, it goes to the propagation mode, where we call G1A if it is under the controlled greenhouse and greenhouse conditions, what also we can call as a multiplication block, and then, G1 and G2, I forgot one there. What typically we call the multiplication blocks that are being established in nursery level. And if test is something positive, it goes through the same process of virus elimination involving a combination of either heat therapy or medicinal-type tissue culture methods, and then it goes into the program. So these are the number of viruses. Don't worry about the small text here. I just want to list the viruses there and the different levels, how many viruses that are being tested. Majority of them, as you mentioned here, leaf holes, red spots, they're all there. There also has, G1 has the requirement of testing for crown gall, which is probably not a requirement in the places like California because they have hotter climate. Crown gall is not a big issue there. So when it comes to the Canadian Grapevine Certification Network, simply to put together the whole long-term certification standard, a nursery or a custom propagator who would like to be involved has to sign a contract with the CGCN, and the grapevine samples will be collected, and the wines, those samples will be collected will be GPS mapped for traceability, and those collected samples will be tested for the viruses of concern that is listed in the standard by approved CGCN lab. Glad to say that CCOVI is one of those approved testing labs for CGCN, and the sample collection virus testing cost at present is shared at 50-50 cost share between the growers and CGCN. And the nursery has requirement to the way 10 cents per wine to CGCN because of certification standards. So to summarize, the Clean Plant program structure, it is always important to have these three components in it. One is research, quarantine, and outreach. Research in terms of what we learn, what we are learning through majorly academic federal government research agencies, where science can improve what we learn about molecular diagnostics, taxonomy, as well as epidemiology of different emerging viruses, and also systematic studies. And this knowledge can be also used in the two other components, outreach and quarantine. Especially when it comes to quarantine, we need to know what are the import and export the regulations, whether any national, domestic, regional surveys are being done for a particular pest or pathogens, and is there any sanitary protocols that this quarantine agencies require. And also it is important what are the latest policy changes that are being made based on the research research knowledge that we gain from. And outreach is also equally important in terms of having this network for the Clean Plant programs through educational programs, both that has to be concentrated both on regional as well as crop-specific knowledge transfer activities. So that I would like to conclude, but first thanks to my funders, there are many there, and thanks to my team. Before finalizing, just show a couple of pictures of virus testing lab. That's Tony, who does all the molecular diagnostic tests. And that's the Grapevine germplasm repository that we are being maintained for close to 3 years now. That's the picture when we just received those materials, but now they look much better. We do test them on a regular basis. This is all possible from funding from the OGWRI as well as CGCN and Brock University to maintain the replica of the Grapevine germplasm repository here at Brock. We do have some bad boys. When you have the certification Clean Plant program requirements for testing different viruses, we do need to maintain virus cultures. So we do have a facility where we maintain the different type of viruses on different grapevine varieties, which we use in our diagnostic methods as a positive controls, as well as we have healthy plants to use as a healthy control, which are really critical when you are establishing the testing platform for Clean Plant programs. So I end with this slide to say it again that we need Start Clean and Stay Clean programs. Thank you very much.
So we've got some time for questions. Any questions from the group? Maybe I'll start with a question. So I was intrigued when you put up that slide about India starting, their own Clean Plant program, and one of their programs is in grape. Knowing that India is expanding their wine industry, the grape program, is it mainly table grapes, or are they also expanding that to wine grapes?
Yeah, the question is India is expanding their Clean Plant program, especially on the grapes, whether it is mainly concerned with the table grapes or wine grapes. To answer that is, yes, table grapes is the big commodity, especially in the southern parts of India, but it's trying to push its boundaries to at least find some microclimatic regions where wine grapes are grown, especially in states like southern Karnataka, the Mysore region, where we do have a few thousand acres of wine grapes. Few wineries are also there, but they're trying to find more ways, especially in the northern parts of the country, to establish just not the production, but some of the universities are actually trying to introduce the programs on focusing on the wine grapes. So yeah, it is trying to expand.
Maybe a potential collaborator down the road, right?
Yeah.
Yeah, very interesting. Yeah.
Similarly, at the top of Outreach, have you worked with someone named Leon Long out of Avignon that runs a Avignon Nursery Plant Program?
Where is that in?
In Avignon, like the south of France?
I'm not sure because we don't really. Sorry, the question is whether I worked with any nurseries in France. We don't really work with any nurseries in France directly because right now our focus is to establish the domestic certification program, the national certification program for grapevines. We want to make sure that at least we are self-reliant, not depending on importing plant material from different countries. But it is also important to know that the regulatory body for Canada, the CFIA, the Canadian Food Inspection Agency, has listed, I think, maybe if I'm wrong, 3 or 4 nurseries in France that are only eligible to export the material into Canada. So anybody who has to get material from France has to rely only on those listed nurseries in France. So I'm really not sure whether that particular nursery that you are mentioning is listed in there or not.
And then the second part of that question is there any people internationally in the business network and Great Nursery plant program that you're working with, and is there any key players in either Ontario, BC, that are working towards starting a nursery?
Well, there are some domestic nurseries that are registered with the Clean Plant the Canadian Clean Plant Grapevine Certification Network, sorry, CGCN. They have listed all the nursery names in their website. There are a couple from here in Ontario, more than a couple here in Ontario. There is one nursery in BC. I think there is one nursery in Quebec as well. There are a lot of interest that is growing among the Canadian nurseries to be registered with the CGCN for certification. Right now, there is no international nurseries that are registered with the CGCN because our main focus is to establish the domestic certification program.
So could you just comment for the group, especially with us going through COVID and difficulties with importing plant material into the country, does that also speak to the critical nature of developing our own domestic clean plant program? Can you just comment a little bit about those challenges?
Yeah, sure. The question is about how the pandemic is influencing providing us the critical need of establishing our own domestic clean plant program. Yes, with the supply chain issues that is created by pandemic, a lot of growers have experienced that, having their materials struck up at the borders because there was no clearance because of the supply chain issues, as well as some of the varieties that we grow probably not a big importance in other parts of the world. So even for us to request particular type of varieties that are important to us, maybe it's not important to other nurseries that are in California or New York. So having to deal with that type of issues, as well as complications with the emerging pests like spotted lanternfly. I heard that somebody has an issue because the quarantine certification doesn't mention that it is inspected or it is free of this particular pest that is emerging, which we don't have it so far. But those type of issues also causing delays in terms of having the material that came into the border, but because of lack of communication, lack of knowledge, lack of clarity between the different nurseries and how the regulatory agencies work here in Canada. So we have a lot of issues in importing those materials. So and also it's all further complicated by we also need to make sure that it needs to be clean of bacterial diseases like crown gall, which probably is not necessary for any material that is coming from California. So people are worried, okay, how do we solve this problem? So unless we have our own certification programs which can support the clones or varieties that are well suited for our climatic soil conditions, pathogens of our interest, I think it is important to emphasize and grow our own domestic game plant program.
Thank you. Any other questions? Oh.
Just in your opinions, how soon can our certification network start to provide clean materials to our nurseries?
It has two aspects of it, how soon it can come into the place. It is in place, and there are two certification standards. One is the verification program, one is the certification program. Verification program is when they look at the viruses of concern, maybe 3 viruses of concern, which is in practice, a lot of nurseries are involved and they're getting tested for that particular 3 viruses. And when it is free of those viruses, they are propagating them, distributing to the growers. The second long-term certification standard, where you need to have, as I mentioned in some of those slides, everything is clean, it has to come from the repository, and the nurseries has to establish its mother blocks and follow the guidelines of certification standards. I think we are on the right track. Maybe in the next couple of years or so, we will be having our own long-term certification standards in place.
Well, Sid, thank you very much for an engaging talk today. And just a reminder for everybody, next week we'll welcome the President and CEO of the LCBO, George Saleas, for his presentation, Uncorking Success, the Business of Wine in Ontario. So we look forward to seeing everybody in person and online next week. Thank you.